Hello future doctors, how are you all? Welcome back to MedSteps YouTube channel. So now we are going to discuss about normal flora in microbiology and some basic things about bacterial structures. In this video we are going to discuss from basics to clinical relevance and management, and there is a what are the main things important for our USMLE and FMGE exams. Okay, so first of all you have to understand the basic thing. You know your body is home to trillions of microbes that help you stay healthy and can also turn against you. So today we are diving deep into normal flora, your microscopic alleles and potential enemies. We will explore what they are and where they live, how they protect you and how they can cause disease. So see this video from beginning to ending, so you will get one idea. Okay, so first of all, what is normal flora? The normal flora means, or you can tell, that is microbiota. Okay, the normal flora refers to the collection of microorganisms. So in this microorganisms, what are the things you can see? Mainly it consists of bacteria, fungi and protozoa, may be present with some individuals. So these things, these microorganisms that live on and inside the human body, that means our body, these microbes are not harmful. Okay, some microbes will be harmful, but this normal flora microbes are not harmful, and under normal conditions, in fact, many are essential for our health. Think of them as permanent residents, helping your body functions smoothly. So functions of normal flora: it will give nutritional support, it will provide useful nutrients. Okay, in nutritional support we have some flora, that is it synthesize essential vitamins like vitamin K, and by vitamin K, vitamin K is produced by E. coli in the gut. Which organism will produce this thing? E. coli in the gut. So this E. coli will produce the vitamin K, so it's very essential vitamin. Okay, so it will help to clot blood. So next one, it will help the main another function is antimicrobial, antibacterial defense. Why? They produce bacteriocins or other antimicrobial compounds, like outcompete pathogens by occupying space and consuming nutrients. Okay, so next one, it will help this normal flora. Main function is the it will maintain the homeostasis. Homeostasis means it will help for maintaining conditions such as pH. Okay, maintain pH means such as lactobacillus in the vagina, which keeps it acidic to prevent pathogen growth. If in vagina, if this any pH value like acidic nature problems, if it came, means that patient can get bacterial vaginosis. So these are the things, and giardia and Yersinia, these are the organisms can cause bacterial vaginosis, vaginal infections. Okay, so that's why this is very important. So main functions of normal flora is nutritional support, antibacterial defense and maintain homeostasis. Okay, so when this normal flora becomes harmful, when means it's maybe misplacement of flora. They can cause infections if misplaced. In misplacement of flora, for example, E. coli. In this E. coli from the gut entering the urinary tract can cause urinary tract infection. Okay, if any E. coli from the gut, if it's entering to the urinary tract infection, then it can cause urinary tract infection. And then skin flora like Staphylococcus, Staphylococcus epidermidis can cause catheter related infections. Okay, this Staphylococcus in epidermidis, you can see this thing in on skin. So while using catheter, okay, that Foley's catheter, that time this bacteria can enter into the urinary tract, so it can cause urinary tract infections. So that's why it's like harmful, it can cause infections. Next one, immunocompromised states. If any patient have like immunosuppressor user like cancer patients or immunocompromised states, then this bacteria, these microorganisms can increase or overgrowth occurs if immunity is low. Example like oral thrush. This oral thrush caused by Candida albicans. Okay, so now we are going to see the very important things, that is sites and common organisms of bacterial growth, or mostly we can see this type of microorganisms in sites. Let's break it down region wise. First of all, I am going to discuss about skin. In skin, we can see Staphylococcus epidermidis. This Staphylococcus epidermidis is like non-pathogenic unless introduced into bloodstream or devices. Next one, nose. In nose, we can see Staphylococcus epidermidis. 25% of people carry Staphylococcus aureus. Okay, can lead to infections. Next one, oropharynx. Oropharynx means mouth plus throat. This oropharynx, we can see like it's like dominated by viridans group streptococci. It causes dental and dental and heart valves infections if they enter into bloodstream. Next one, dental plaque. This in dental plaque you can see like Streptococcus mutans. It's like major player in dental caries. Next one, colon. Colon means large intestine. In large intestine, we can say like 99% of anaerobes like bacterioids, in that 99% of GI tract bacteria, and E. coli also present can cause infection if displaced. Next one, vagina. In vagina, we can see dominated by lactobacillus. It produces lactic acid. It inhibits pathogens. Okay, like it inhibits pathogens, or you can tell it suppresses the pathogens. Next one, we can see the urethra. In urethra, next we can see the urethra. In this urethra, mostly you have to focus on also harbors a small population, usually harmless unless introduced into bladder. Okay, so special note here, main thing is in neonates, neonatal colonization, we can see like fetal GI tract will be like very sterile. GI tract of fetus is sterile. So colonization begins at birth. When this colonization will happen, this colonization will begin after it begins at birth. First of all, we can see like vaginal delivery. In vaginal delivery, that baby is exposes neonates to mother's flora, good for immune development. And we can see cesarean babies lack this exposure, resulting in delayed flora stability and potentially they are higher risk of immune disorders. Okay, so in clinical applications, what are the things important in this neonates? Means diagnostic cause. In diagnostic cause, we have to know the normal flora helps interpret cultures. Example like S. epidermidis in blood might be contaminant or pathogen depending on context. Source of infection. In immunocompromised patients, normal flora can cause like sepsis, urinary tract infections, or endocarditis, or etc. And antibiotic associated problems like broad spectrum antibiotics can disrupt normal flora, leading to super infections like clostridioids, difficile colitis. Okay, so how we can prevent this type of organisms and pathological conditions? Means probiotics. Probiotics will help to restore the flora, especially during or after antibiotics. And we can see like infection control in hospitals, like minimizing the or reducing the use of catheters, and maintain hygiene in ICU patients to reduce flora associated sepsis. In neonatal care, mostly we have to focus on consider maternal microbiota's role, encouraging breastfeeding and skin to skin contact. And immunocompromised patients, we can say like monitor for overgrowth, example candida, gram negative rods, and use target antimicrobial to prevent flora related infections. So here main thing is normal flora is silent bodyguard but can becomes enemies also, crucial for vitamin synthesis, immune development and pathogen resistance. In understanding flora helps in diagnosis, infection control and preventive medicine. Okay, so these are the main things in normal flora. So now we are going to discuss about very important and high yield topic, that is bacterial structure. In bacterial structure, based on the part of the bacteria, we have to discuss, we have to go little deep in this video. Okay, so have you ever wondered how the structure of bacteria helps us identify them, target them with antibiotics and understanding their behavior in infections? So today, so in this video we are discussing about this clinically, what are the structures will clinically important for this microbiology. Okay, so we are going to discuss from basic to clinical relevance. So now first one, bacterial structure. In bacterial structure, first one, bacterial shape. This is the shape is key for identification. In this bacterial shape is one of the first ways we identify the bacteria, seen through staining and light microscopy. Like it depends on how the bacterial cell wall is assembled. Okay, so here we have some types of shapes, like first one, rods and round means we can call that is coccus. Next, rod like structure, that is bacillus, and spiral. Okay, so you can see this image. First one, bacilli. Bacilli means rod shaped, example E. coli. Okay, next one, cocci. Cocci means it's like coccus, it's like round, example like streptococcus and staphylococcus. Next one, spirali, or you can call spirillum. It will be like corkscrew, like example Treponema pallidum. Okay, so first of all we have some special patterns also, that is diplococci. Diplococci means paired, or example you can say like Neisseria, and chains. Chains means example streptococcus, and clusters like grape like clusters, example staphylococcus. Okay, so next one, pleomorphism. This some bacteria will be like species like pleomorphic, also known as pleomorphism. Pleomorphism means some bacteria like bacteriodes are pleomorphic, meaning they can change the shape based on based on disease. In clinically important is penicillin and beta lactams. It inhibits cell wall synthesis and this can alter bacterial shape, making shape not only diagnostic but also a sign of antimicrobial activity. So first we are going to discuss about nucleus. So what is nucleus? Bacteria don't have a true nucleus, they have a nucleoid containing a single circular double stranded DNA molecule, the bacterial chromosome. Okay, so in plasmids, extra chromosomal circular DNA can replicate independently, often carry antibiotic resistance genes or virulence factors. So in plasmid, what's the main importance for plasmid? Plasmid means these plasmids are how bacteria share resistance via horizontal gene transfer conjugation, a huge problem in hospital acquired infections. Next one, cytoplasm. In cytoplasm we can see like no organelles, we can see like no mitochondria, no endoplasmic reticulum and no Golgi apparatus. Okay, so it contains, this cytoplasm contains ribosomes and storage granules. So ribosomes, in these ribosomes they are the site of protein synthesis in the cell. the ribosomes. If you are seeing this image here, you can see here some of the ribosomes. This ribosomes is mostly we have to think about, it's like site of protein synthesis. Okay, and another thing, it's prokaryotic ribosomes, we can say like 70 years, in 70 years we can say like 50 years plus 30 years, and eukaryotic ribosomes will be 80 years. Why this is important? The antibiotics like tetracyclines and aminoglycosides, it's mostly target the 70s ribosomes, selectively killing bacteria without harming human cells. Okay, so next one, cell wall components, or you can see cell or cytoplasmic membranes. Here this is gram positive and gram versus gram negative. You can see this image here. In gram positive, we can let's compare the both type of using structural based breakdown. You can see this image, based on that we can differentiate. First one, I am going to discuss about gram positive. In this gram positive, you can see in gram positive bacteria, you can see here thick peptidoglycan wall. Okay, you can see here, you can compare this thing. Here in gram negative, you can see only one layer of the peptidoglycan, but in gram positive, you can see here 1, 2, 3, triple layer thick peptidoglycan wall. You can see here in gram positive. Next one, lipoteichoic acid. This lipoteichoic acid is the unique in, mostly you can see in only gram positive. It's like mostly you can see mainly you can see in gram positive. Next, no outer membrane, we can't see any outer membrane compared with gram negative. In gram negative, you can see endotoxin and lipopolysaccharide and porins, mostly you can see in outer membrane. Okay, but in gram positive, no outer membrane. Next one, gram negative bacteria, thin peptidoglycan, already discussed, outer membrane with lipopolysaccharide, or you can call endotoxin, and porins, protein channels for molecule entry, and periplasmic space contains beta lactamases and antibiotic destroying enzymes. Next, common structures. In these common structures, mostly you can mostly you have to focus on flagella. This is the flagella or flagellum. This flagella is help for motility of the bacteria. Next one, you can see here pili, that small fine-like structure. This pili is help for adhesion and DNA transfer. Okay, next one, capsule. You can see here, this is the capsule. This capsule for immune evasion, it will help for immune evasion. Next one, cytoplasmic membranes. The cytoplasmic membrane is main for passing the molecules. Okay, next, clinically what important with this image means the gram staining helps guide empirical antibiotic therapy, and gram negative bacteria are harder to kill due to lipopolysaccharide and outer membrane. So this beta lactamase inhibitors are added to antibiotics to overcome resistance. Okay, so next, cytoplasmic or cell membrane functional powerhouse. So it's made of phospholipid bilayer. This cytoplasmic membrane is made by phospholipid bilayer. Bilayer means two layers made by phospholipid. So it lacks sterols except in mycoplasma. It's embedded with enzymes and penicillin binding proteins. So main functions will be like, it will help for selective transport. Main function will be like selective transport. This selective transport mostly we can see here diffusion and active transport. Diffusion and active transport. Okay. Next one, enzymes excretion and excretion of extracellular enzymes. So this enzymes excretion, it's like digestive enzymes and antibiotic degrading enzymes. Next one, respiration. In respiration site, no mitochondria. Here we can't see any, no mitochondria, and so the cytoplasmic membrane handles it. So next one, cell wall biosynthesis. In the cell wall biosynthesis, you can see enzymes and lipid carriers assemble peptidoglycan units here. Okay, so this here you can see this Clinically, what importance here based on this thing means that here you can see this PBS are targeted by beta lactam antibiotics and mutations. This here you can see, okay, based with this image I will try to explain this thing. Here clinically what important, why we have to know about this cytoplasmic membrane. Here you can see here penicillin binding proteins. These penicillin binding proteins are targeted by beta lactamase locations or beta lactam antibiotics. Its mutations in penicillin binding proteins like resistance, it's seen in MRSA. And this respiratory function of membrane is targeted by oxidative damage on certain antibiotics. Okay, so mostly we have to focus in exam view for FMGE and these things. We have to focus on based on gram positive and gram negative. If any person asking about thick peptidoglycan, you can tell gram positive have thick peptidoglycan and is targeted by penicillin. Next one, lipopolysaccharide or endotoxin. It's mostly you can see in gram negative and targeted by immune activation. Next one, outer membrane. This mostly outer membrane we can see in gram negative and targeted by antibiotic barrier. Next one, penicillin binding proteins we can see in gram positive and gram negative and mostly the targeted by beta lactams. Next one, periplasmic space we can see in gram negative and targeted by beta lactamase inhibitors. Next one, ribosome. That ribosome 70S we can see in gram positive and gram negative and targeted by tetracyclines and aminoglycosides. Okay, so in clinically high yield clinical things mostly we have to know, that is Neisseria. Neisseria will be gram negative diplococci, the shape and stain. Okay, and staph aureus will be like gram positive cocci in clusters. It's important for skin device related infections. Next one, E. coli. The E. coli will be gram negative bacillus from the gut, and mycoplasma will be like no salval. We can see we can't see any salval, so no salval, and it's resistant to beta lactams. And the bacillus anthracis is like we can see rod shaped gram positive spore formation and bioterrorism relevance. Pharmacologically, these things are very important because gram positive and negative status, presence of penicillin binding proteins and ribosome type and determines drug of choice, and knowing the structures helps to pick between beta lactams, microlides and aminoglycosides antibiotics. Okay, so always correlate with gram stain plus clinical presentation before starting antibiotics. Okay, so use broad spectrum antibiotics wisely can lead to super infection. So we have to use antibiotics that targets penicillin binding proteins like ribosomes or membrane depending on structures, and vaccines like pneumococcal target and capsule structures of bacteria. Okay, so next one we are going to see the plasmids. Now plasmids. What are plasmids? Plasmids are small circular non-chromosomal double-stranded DNA molecules that are self-replicated capability the plasmids have. Okay, so they are extra chromosomal, meaning they exist outside the main bacterial chromosome. It capable of self-replication independently of chromosomal DNA. The functions of plasmids: it's carry non-essential but beneficial genes for antibiotic resistance, for example beta lactamase enzymes, and virulence factors like toxins, adhesions, immune evasion mechanisms, and fertility factors like F plasmid involved in conjugation. The plasmids are the main reasons for multi-drug resistance in hospital, especially in E. coli and Klebsiella and Pseudomonas. So here how we can manage these things means we can use combination antibiotic therapy in suspected MD or infections like multi-drug resistance, and avoid unnecessary antibiotic used to prevent plasmid selection pressure. Okay. So next one, transpositions. What are transpositions? Transpositions are segments of DNA that can move from one location to another between chromosomes, plasmids, or both. It unlike plasmids, they cannot self-replicate. So in functions, mostly we can say like it carries genes for antibiotic resistance, enzymes, toxins, or metabolic functions. It can disrupt normal bacterial genes, altering like virulence or metabolism. The key drivers of mutation and evolution in bacteria. The clinically importance is a resistant bacterium may have acquired transposition mediated vancomycin resistance. For example, you can say like vancomycin resistance enterococcus. You can use this bacteria will be resistant, so we can use combination drugs, antibiotic drugs. Next one, cell wall. Cell wall is also known as the bacterial armor. The general features of the cell wall means it surrounds the cytoplasmic membrane. It composed mainly of peptidoglycans. It gives rigidity and shape of the bacteria. Its major target for antibiotics like penicillin and cephalosporins. Okay, so here gram positive cell wall. In this gram positive cell wall, you can see here thick peptidoglycan. This gram positive cell wall. Here you can see peptidoglycan. This will be like thick peptidoglycan layer up to 40 sheets. It contains lipotechoic acid. It serves as a surface antigen. For example, Staphylococcus aureus. Clinically, the gram-positive bacteria are more susceptible to penicillin unless resistant. Example like MRSA. MRSA means like methicillin resistant Staphylococcus aureus. And lipotechoic acid can trigger an immune response in the host. Okay, next one. Next you can see here gram negative cell wall. The gram negative cell wall will be, you can see in gram negative mostly you can see here thin peptidoglycan 5 to 10% of wall, and outer membrane contains like lipopolysaccharides, LPS, or specifically lipid A. It acts as endotoxin. And you can see periplasmic space contains enzymes like beta lactamase. For example, you can see like E. coli and Neisseria and Pseudomonas. In clinically important will be like its gram negative infection can cause septic shock due to lipid A endotoxin. The periplasmic space protects beta lactamase enzymes and antibiotic resistance. The management strategy in severe gram negative sepsis use carbapenems or beta lactamase inhibitors. And we have to monitor for signs of endotoxemia like fever, hypertension, and disseminated intravascular coagulation. Okay. In special cases, no cell wall. In special cases, we can say like mycoplasma. In mycoplasma, we can say like no cell wall. Okay, and only a cell membrane with the sterols appears pleomorphic and no fixed shape and resistant to cell wall inhibitors like penicillin and cephalosporins. The key organisms will be like mycoplasma pneumonia. Okay, how we can treat? We can treat with antibiotics that target ribosomes like macrolides, azithromycin, tetracyclines, or fluoroquinolones. Okay. So plasmid means the main function will be carry the resistance, virulence genes. Transpositions means it's like mobile DNA, mutations and resistance. Gram positive cell wall means thick peptidoglycan, lipotechoic acid. Gram negative cell wall means lipopolysaccharide, endotoxins, and beta lactamases. Mycoplasma means no cell wall. Okay, so these are the things. So next we are going to discuss, we are continuing with capsule and related structures. Capsule and related structures. Here, what is the capsule? The capsule is a well organized extracellular polysaccharide layer tightly attached to the bacterial cell wall, and we have some exceptions like Bacillus anthracis has a polypeptide capsule made of the glutamate. Okay, so this it can visualized by differential staining, appears as a halo around the bacterium under the light microscope. So the related structures, in this related structures we can see like slime layer. Okay, the slime layer will be like loosely attached unorganized version of the capsule. Next one, glycocalyx. The glycocalyx is like a mesh like polysaccharide matrix extending outward, important for adhesion. Next one, functions. The functions will be like first one, antiphagocytic. Antiphagocytic means it inhibits engulfment by neutrophils and macrophages. Next one, virulence factor. In this virulence factor, it increases pathogenicity, example Streptococcus pneumonia, and protection against the antibiotics, desiccation and host defenses. Next one, adhesions. The adhesions helps bacteria stick to surfaces, especially in biofilms. Example, we can see Streptococcus mutans adheres to tooth enamel using its glycocalyx. Here the clinical importance is its capsulated bacteria are more virulent. Examples like Streptococcus pneumonia, Neisseria meningitidis, Haemophilus influenza type B, Klebsiella pneumonia, and Bacillus anthracis. And we can see like Q-lung reaction used in labs to detect capsules, and we can see like vaccines based on capsular polysaccharides. Example, hepatitis B, pneumococcal and meningococcal vaccines. Next one, we are going to discuss about appendages. In appendages we can see flagella and pili. First we are going to discuss about flagella. The flagella will be, we can see the structure and composition will be like long whip-like appendage made of flagellum, or we can tell H antigen. It have like number and location varies. First one, monotrichos. This monotrichos will be like single flagellum. You can see here this is single flagellum. Next one, example we can say like Vibrio cholera. The Vibrio cholera we can see that bacteria will be like monotrichos. Next one, lopotrichos. In this lopotrichos like multiple flagellum you can see here, you can see this image, you can appreciate that thing. So lopotrichos, multiple flagella at one pole. Next one, amphitrichos. Amphitrichos means flagella at both ends. You can see here both ends have flagellum. Next one peritrichos. Peritrichos means all around you can see this flagellum. Example like E. coli. So this main function of the flagellum means it will help for motile bacteria for motility. Example Proteus mirabilis, it shows swarming motility on culture media. The flagellum is highly antigenic detected as the H antigen in serotyping, example Salmonella. Next we are going to discuss about pili. Pili, or you can call fimbriae. It will help for attachment and DNA exchange. Here this structure will be like the pili and fimbriae will be like thin hair-like projections made of pilin protein, and it's like shorter and thinner than flagella. The types of pili and fimbriae will be like types and functions will be like adhesion pili, fimbriae. It help bacteria stick to host cells and surfaces, like Neisseria gonorrhea uses pili to adhere to urethral epithelium, resisting flushing by urine. The sex pilus. The sex pilus will be like long singular pilus used in conjugation. It's transfer of plasmid DNA. Next one is clinically what's important of this pili fimbriae means it will help for virulence factors critical for initial colonization of host tissues, important in biofilm formation, recurrent urinary tract infections and antibiotic resistant spread. The bacterial spores or endospores. The bacterial endospores are we can call this is survival specialists. What are endospores means? It's like dormant, non-reproductive, highly resistant forms produced by some gram-positive bacteria. The genera will be like Bacillus and Clostridium. One vegetative cell forms and or one spore and vice versa. The sporulation will be like formation of spores triggered by harsh conditions, nutrient depletion, extreme pH, heat or dehydration. The bacterium will be like encloses part of its cytoplasm with DNA and adds a thick cortex and a resistant spore coat, and it remains dormant until favorable conditions return. The key structures of inside the spores means DNA, and we can see ribosomes and dipicolinic acid like calcium major contributes to thermal resistance, low water content resistance to drying. Okay, so in germination it's returned to life with when conditions improve, spore absorbs water, spore coat breaks down, a new vegetative cell emerges within 15 minutes. So the disinfection tips will be like spores are heat resistant, so 60 degrees Celsius kills vegetative cells. The 121 degrees Celsius moist heat for 15 to 20 minutes autoclave, it's required a kill spores. In lab diagnosis will be if you are suspecting spores, when recurrent hospital infections, organisms surviving high heat process, no growth unless special anaerobic conditions used. So the capsule will be like the capsules will be like antiphagocytic adhesions. Clinical examples like streptopneumonia and Haemophilus influenza. The flagella will be like motility and Haemophilus antigen. The clinical examples will be like E. coli and Salmonella. The structure will be like pili structure, will help function will be like attachment and DNA transfer. The clinical example will be like Neisseria gonorrhea and conjugation. The spores, the function will be like dormancy and resistance. The clinical example will be like Clostridium difficile and Bacillus anthracis. Okay, so these are the things is main important about bacterial structures. So you can see here this is the spores. In the spores we already discussed about vegetative cells, then developing spore coat and the mother cell and spore coat and free endospores. So in this video we discussed about normal flora and bacterial structures. In every bacterial structures we went deep and we discussed many things and clinical relevance and what are the functions and some examples of bacteria also. So in next video we are going to discuss about the structures and functions and chemical compositions. Okay, so these are the things for this video. So this is my first video of microbiology. So I am following the first aid guide. If you have any doubts you can ask me in comment section. So till now you didn't subscribe my channel, like, share and subscribe. If you are if this video is help you for studying purpose, so let's subscribe my channel and share to your friends. Take care, bye bye, we will meet in our next video. Till that time,